Welcome back. In this session, we will begin a new lesson, that is Principles of Inheritance and Variation. In the previous sessions, we have discussed about reproduction in plants and reproduction in animals, wherein we learned that a parent produces an offspring similar to itself. In the case of asexual reproduction, the parent as well as the offspring are almost replicas, so they are called as clones. Whereas in the case of sexually reproducing organisms, the basic body design of the offspring is similar to the parent, but there are obvious differences between the parent and the offspring. So, where do these variations or differences come from, and how are these passed from the parent to the offspring is what we will be discussing at length in this lesson, inheritance and variation. Let us understand these two terms. Inheritance, or sometimes it is also called as heredity, is nothing but heredity is the study of how characters are passed from the parent to the next generation. So it deals with the study of how characters are passed. So passing of characters. We call the characters as traits. These are observable features. For example, the hair is black. Is it straight or is it curly? The color of the eye, we can observe it. So these are called as characters, features or traits. So how are the traits passed from the parent to the next generation, that is the offspring, is what is called as inheritance. And of course, in sexually reproducing organisms, the parent and offspring are not exactly replica copies. They have certain similarities. Of course, they have their differences. Variation is nothing but the degree of change between the parent and the offspring. To what extent the parent and offspring are different is what is called as variation. So that is the meaning of these two terms. How are the traits passed and what is the amount of change between the parent and the offspring. The branch of biology which deals with the heredity and variation together is genetics, which studies about genes, how these genes are passed from the parent to the offspring, and what are the variations present between the parent and the offspring comes under genetics. At present, we know that every character or trait is due to the gene. Genes are hereditary units that reside on the chromosome. Chromosomes are present inside the nucleus of a cell. When a cell divides, the chromosomes are also equally distributed between the two cells. Therefore, the basic information that is present in one cell is distributed to another cell. So, such basic hereditary units are called as genes. But our ancestors had no idea about genes or chromosomes. Still, they practiced genetics unknowingly. For example, around 8000 to 1000 BC back, our Indians, they developed new breeds of cows from wild cows. with the help of artificial hybridization. Also, development of different varieties of rice and certain crop plants, etc. All which were practiced by our ancestors with the help of artificial hybridization and crossing, wherein they used to observe the features or traits of a plant or an animal. Then, based on their requirement, they used to hybridize them, or they used to cross these two varieties, and in turn, they used to obtain both the favorable traits in a single organism, which was now called as a hybrid. So that is how different types of crop plants and commercial plants were developed by our ancestors. Similarly, the animals, domestication of wild cows, domestication of wolves into dogs, all these are a result of this. So that is what is genetic and is factors. But they did not have any idea as to what was the reason for these traits being passed from parent to the offspring, or what was the reason for differences between the parent and the offspring. Only in the 19th century, the detailed study of genetics started through Gregor Johann Mendel. He is called as the father of modern genetics. Gregor Johann Mendel was an Austrian monk. He was interested in biology as well as mathematics. Gregor Johann Mendel, or simply Mendel, he carried out detailed experiments on the plant garden pea. The pea plant, the scientific term nomenclature is Pisum sativum. So he carried out experimentations on this plant for a period of 7 years, from 1856 to 1863, asking fundamental questions of genetics. How are traits passed from one plant to another plant, and what is the cause of variation? So Gregor Johann Mendel chose this particular plant for his experiments for a couple of reasons. Number one, they have a relatively short life cycle. Once the plant grows, it flowers within 30 days. Within a month, you can obtain a new generation, which is very, very important for experiments of genetics. Not only that, these are self-pollinating plants. They undergo self-pollination, but they can be easily cross-pollinated, and they show contrasting characteristics or contrasting traits, which are very important for this particular study. Contrasting trait meaning features that are opposite to each other. So Gregor Johann Mendel found such 14 contrasting traits, or 7 pairs. You can see at the diagram, trait, for example, height of the stem. Height of the stem has got two contrasting traits. If the stem is tall or whether the stem is dwarf. So these are contrasting traits opposite to each other. If you look at the pod shape and pod color, whether the pod is inflated or is it wrinkled, is it yellow or is it green. If you look at the shape of the pea, the fruit, whether once again is it yellow or green, and whether the pea is round or wrinkled. Not only that, we also look into the color of the flower. Flowers could be either violet or white, and the position of flower on the stem. Certain flowers were axial, that is on the sides in position, whereas certain plants had flowers on the top, terminal position. So these 7 pairs of contrasting traits were observed in the Physacetum plant. All these were helpful in the study of genetics. Before we move on to discuss about Mendel's genetic experiments, we have to understand a few terminologies so that we can easily grasp Mendel's experiments. Trait, as you already know, is nothing but a feature or a character. For example, eye color is a trait, or height. So these are characters or features. Phenotype. Pheno means what is observed, what we see. So it is a observable feature. For example, let us say stem height. Is the stem tall or is the stem dwarf? So that is a phenotype. So color of the eye, is it black, is it blue, is it brown? We can see it. So that's what is called as a phenotype. It's an observable feature. Genotype. Genotype is nothing but the genetic makeup or genetic constituent. We know that every trait is controlled by the gene. So what is the genetic makeup of height of the stem, or what is the gene that is responsible for the color of the eye? So the genetic makeup or genetic constituent of the trait is its genotype. Now we know that in sexually reproducing organisms, they are diploid, meaning there is a copy of every chromosome. So chromosomes are present in pairs. Take for example human beings. Human beings have 46 chromosomes, out of which there are 22 pairs of chromosomes, and then there are 2 sex chromosomes. 22 pairs of chromosomes, wherein 11 chromosomes are present, plus there are copies of it. So a diploid organism carries a copy of every chromosome that it has, other than the sex chromosome. So there are 22 chromosomes and 22 copies of these chromosomes. So that is a diploid organism. So let us say this is chromosome number 1 in a human cell. It will have another copy of itself, and they are exactly same. They contain the same number of genes, the same type of genes at the same positions. Let us say there is a gene that is present here that is responsible for eye color. The exact same gene will be present in the same position in the copy also. So these are the genes. So genes are present in twos. This we call it as alleles. So one of the gene will be responsible for, let us say, tallness. Another gene will be responsible for dwarfness. So the contrasting features are present on the chromosomes. So alleles are basically that alternative forms of the gene present on homologous chromosomes. Both are present on the same position. Alleles are alternative forms of the gene present on homologous chromosomes. We can name these alleles using capital letter and small letter. So there should be a way of identifying these alleles. So we use this kind of nomenclature. For example, let us say this is a tall plant and this is a dwarf, short-heighted stem. So let us say we use the alphabet T. So tall plant will be represented by T, whereas the dwarf plant will be represented by the small case of the same alphabet. We should not use the alphabet D. Why? Because these are alleles. These are the alternative forms of the same gene. The gene is for the height. So the tall allele will present in this chromosome and the short allele will present on this chromosome. So that is how we identify the alleles using alphabets. Now there is also a rule of thumb to be followed when we choose these alphabets. In this case, we could have chosen alphabet D also, and D for tallness and D for small d for dwarfness, but we don't do that because the reason is the alphabet that we choose to mark or recognize the gene, it should be a dominant trait. The trait that is dominant, the alphabet will be taken for that trait. What is a dominant trait? What is a recessive trait? A dominant trait is one which is seen in majority of the population of that species. For example, if you look at our Pisum sativum plant, that is garden pea plant, which Mendel used for experiments, majority of the plants are tall. Only few, 25% or 30% of the population is dwarf. Therefore, based on the stem height, tallness is a trait that is dominant. It is seen in most of the population, whereas dwarfness is seen only in few members of the population. So the letter that we use to mark the allele is based on the dominant trait. So dominant trait is seen in most members of the population. Trait that is seen in most members of the population. And this recessive trait is seen in less members of the population. Why is this? This is because the dominant trait does not allow the recessive trait to show up. It will suppress or it will mask the expression of the recessive trait. We already know that sexually reproducing organisms are diploid. So take this example of tallness. there are two alleles for the same gene. One is a capital allele, one is a small t. Stem height is represented by capital T, small t, where capital T is for the dominant allele and small t is for the recessive allele. When dominant allele and recessive allele are present together, the dominant allele will suppress the expression of the recessive allele. Because of that, the recessive allele cannot express itself. Therefore, most of the members will show the dominant trait, that is tall. If you look at the 7th pair of contrasting characters, you will find that the round seed is dominant over wrinkled seed, yellow seed is dominant over green seed, inflated pod is dominant, and green pod is dominant. Flowers are axial in position and violet colors are dominant, because these are seen in majority of the species. Why? Because the dominant allele does not allow the recessive allele to show. So it is a trait that is not expressed in F1 generation, whereas we can say that dominant allele is the one, the trait that is seen in the F1 generation. This is what Mendel did when he carried out a cross between a tall plant and a dwarf plant. He found that the plants produced from the seeds of this cross all are tall. So tallness is a dominant trait, whereas recessive trait is not expressed. Zygous means a zygote. A zygote, you know, is formed when the male gamete and the female gamete fuse. So homozygous condition is where both the alleles are same in a hybrid. For example, capital T or small t, small t. In this condition, both the alleles are tall alleles, and here both the alleles are dwarf alleles. So, in the hybrid condition, if both the alleles are same, it is called as a same zygous condition. Whereas in a heterozygous condition, the alleles in a hybrid are different. So what would be the case? Different means there is one dominant allele and then there is a recessive allele. So this is what is called as a heterozygous condition, where both the alleles are different. If both the alleles are same, it could be tall, it could be dwarf, does not matter. Alleles are same, it is homozygous condition. The last term is hybrid. When you cross a tall plant with a dwarf plant, you get, we will explain this in detail. With a dwarf plant, what happens is, what we get is called as a hybrid. It's a hybrid because it contains alleles from both the parents.